All right. So welcome to the November Cascade Mycological Society meeting with an invited speaker. Our speaker tonight is Charlie Bruder, sorry. And I'll just read from the website that Charlie is going to talk about the domestication of fungi and food systems and a basic history. Um, the story of plant and animal domestication is vast, but Charlie Bruder's interest is unparalleled. In this talk, Charlie will cover a basic history of edible plant and animal domestication and discuss the implications of domesticating fungi for food systems. Conventional means of breeding fungi as well as genetic engineering / CRISPR will be discussed. Both biological and cultural reasons surrounding domestication will be explored. And a little bit about Charlie's background. He's an aspiring mycologist here up at OSU. He's passionate about foraging and community-based agriculture. He's certified in permaculture design, and he aims to bring an ecological perspective to the rising popularity of mushrooms in our food systems. Currently, he works in the wood decomposition lab, where he helps maintain one of the largest cultural libraries of native fungi in the Pacific Northwest. Charlie practices elements of Korean natural farming and other organic methodologies in his own farming and gardening, hoping to increase soil microbial diversity in agricultural systems. Please welcome Charlie Bruder.
Thank you, everybody, so much for coming. I know it's a pretty rainy night out, and I really appreciate everyone making it out, nevertheless. So I wanted tonight's talk to be kind of a basic history. We're going to go to a few different places and give a good, solid foundation for the topic at hand. We're not going to go too deep into any one thing, but really appreciate you coming, and let's get into it. Okay, all set? Okay, we're going to go ahead and get started. So today's talk is going to be covering the domestication of fungi. And so I kind of wanted to center us here on this timeline with some very rough dates of the evolutionary history of the Earth. So we kind of had life emerging on Earth about 3.5 billion years ago. And some key points would be that aquatic plants emerged around 1 billion to 700 million years ago, and fungi shortly after, around 680 million years ago. Terrestrial plants and aquatic animals and mammals followed, and the dinosaurs were right about 250 million years. Here we go. Okay, but humans were actually only emerged around 230,000 years ago. So compared to the rest of life on Earth, humans are very new to this planet. We're a very recent species, and most of our history is completely unknown to us. It's pretty amazing to me that despite the fact that we believe that we're so intelligent and that humans have all these abilities to share knowledge and information with each other, we've forgotten a huge portion of our past. We know that humans emerged in Africa around 230,000 years ago. And up until around 10,000 years ago, we don't really know much about our history. In this middle section where not much is known, we do know that current members of Congress were born sometime around here. But besides for that, the rest is still pretty murky. But around 15,000 years ago, something interesting started to happen, which is that humans started domesticating other organisms. And we'll get into exactly what that means. But the big picture here is that humans haven't been around for very long. And even for the time that we have been around, we've been domesticating organisms for just a very small fraction of that time. So first of all, we need to discuss what is a symbiosis. Well, oftentimes you hear a symbiosis as being a beneficial relationship. That's actually not true. A symbiosis just means two or more organisms living together. And symbiosis is a spectrum that can range from parasitism to commensalism to mutualism. And so a parasitism is a relationship where one organism benefits and the other is harmed. A commensalism is a relationship where neither organism is harmed, and a mutualism is a relationship where both organisms benefit. And so domestication is a type of mutualistic relationship that arises among two or more organisms. And so this really good definition I found here, it's important to note that domestication is very difficult to define, even scientifically. For a long time, it's been very difficult for anyone to define exactly what domestication is. But a really good definition I found is that domestication is a coevolutionary process that originates from a mutualism. It features a domesticator, which is us as humans, and a domesticate, which could be a plant or a mushroom or an animal. The domesticator constructs a hospitable environment, And the domesticate produces goods or services, just like the food that we have here, these vegetables. And this leads to an increased amount of fitness. And this term fitness in an evolutionary sense means greater strength or greater ability to reproduce. So we help these plants reproduce and have successful lineages over generations. And in return, they feed us, allowing us to be more reproductively successful. And so if we look at the domestication of animals, this started, actually, animals were the first organisms that humans domesticated. The first known domesticated organism was the wolf, which is now the dog. And that occurred somewhere between 30,000 to 15,000 years ago in Eurasia. And the domestication of animals was really important for humans for a number of reasons. Obviously for food, raising cattle or other animals to actually slaughter and eat their meat was important. But more importantly, before we had transportation, animals served as a way of getting around, as a way of carrying things that were heavy. So this was a key step in allowing humans to move across the world. There's actually one theory that proposes that the reason that people arrived in the Americas from the Old World or from Europe and not the other way around, why people didn't arrive in Europe from the Americas, was because the megafauna that lived in the Americas were llamas and alpacas, and they were very difficult to domesticate. So they got domesticated later, and as a result, humans didn't have the technology to develop the wheel and more methods of transportation, which would have allowed them to eventually build boats and stronger machinery to get to Europe. So as we look at human history, the domestication of animals was a really key component. And so the domestication of plants is really interesting because compared to animals, where we had only about 40 species of animals that humans have domesticated over tens of thousands of years, humans have domesticated more than 1,000 plant species. So this is really our closest relationship as far as a human to other kingdom domesticated relationship. And if we look at just a few examples, some that are really amazing to me are the transformation of teosinte, which is a grass from Meso and South America, into corn. So corn was originally this very small grass that produced these very hard seed heads. And humans domesticated it to produce these really huge cobs that we can eat and turn into food and process much more easily. And this was the basis for many of the civilizations in the Americas prior to colonization. We also have the rose, so plants can be domesticated for shape and appearance. It's really interesting to me that we take plants and domesticate them and put a lot of energy into them, not just for food, but also just to enjoy life more. We have carrots, which go from just this tiny root to a beautiful carrot like we have today. And we have melons, which are especially close to me because I worked in a melon breeding project for a year. And so we have our original wild melon up there, and then we have modern cantaloupes, which can range all these different shapes and sizes. There's hundreds of varieties, tons of different colors, and I just love them. But plants are really the basis of modern agriculture. They're what feed us, they're what feed our animals so that we can eat meat, and they're the basis of most of the calories that humans produce. But so why is it that we've domesticated so many plants and so many animals, but the domestication of fungi is not really a discussed topic? When you hear about a domesticated organism, a mushroom isn't really what comes to mind. And mainly, the reason that I think this is that humans just haven't been growing mushrooms for very long. We've been cultivating actual macrofungi or mushrooms for maybe about 2,000 years. Depends what part of the world you're in. In Europe, we've only been doing it for about 300 years, and that started with the button mushroom being cultivated in France. And in the Americas, we're just getting started with the exception of a few that we'll get into. But in modern Western the United States, we haven't been growing mushrooms for very long. We have a big industry in button mushroom production, but the amount of species we grow is pretty limited. And the amount of species of mushrooms that are cultivated in general is pretty small. You have less than 10 species that comprise most of the market. Shiitake is a big one, button mushrooms, obviously. And then you have other specialty products. Asia, there's some like enoki that go into ramen and soups. But we don't really have that many mushrooms that are cultivated as a whole. More important than the domestication of macrofungi and mushrooms for fungi is the domestication of microorganisms. So fungi aren't just mushrooms. They can also be microbes such as yeasts or molds. And these are super important for a variety of different cultures. The organism that actually creates soy sauce is a mold called Aspergillus oryzae. And it's what's known commonly as koji. And so this ferments rice into more available sugars that can be fermented into sake to make tamari. We also have a yeast, Saccharomyces, that ferments wheat and other sugars into beers, wines, ciders, and it also makes biofuels. It's important in industry. And then in Europe, we had different cheese molds like penicillium that fermented milk into edible cheeses that helped them preserve longer. So microorganisms, in the case of fungi, are probably more important than the macrofungi and mushrooms as far as domestication goes. Especially important is that they've also been used to produce antibiotics, so most famously penicillin. Completely changed the course of human history as far as how many humans have survived and how many humans don't die of infections. We also have some fungi like trichoderma that are important for making the enzymes that are in your laundry detergent. So this fungus is grown in big bioreactors and the enzymes are filtered out and put into your laundry detergent. I think it's really interesting that these three crops make up most of human food consumption. So wheat, rice, and maize, three grain crops, feed more than 60% of human caloric intake every year. But isn't it interesting that all three of these crops have an associated fungal microorganism that is key to its digestion by humans? So for the case of wheat, we have Saccharomyces or brewer's yeast. That is what turned wheat into bread and wine and allowed it to be consumed and preserved. We also have Aspergillus oryzae or koji in the case of rice, which forms the basis of Japanese cuisine. And for maize, we have corn smut, or Eustolago metis, which is a very interesting fungus. So let's talk about corn smut, also known as huitlacoche. It's a traditional food in Mexican and Mesoamerican cuisine, and it's usually served within corn tacos. So what this actually is a fungus that parasitizes corn and grows on it and converts some of the starches and other starches and sugars in the corn into proteins that are more valuable. So people in the Americas knew this. They knew that corn smut was nutritionally valuable, and they selectively inoculated their corn plants with it in order to ingest it. And it has a lot of cultural significance and nutritional significance, and I think this is why it became part of this whole foods diet that took place in Mesoamerica. So people talk a lot about how mushrooms are good for you, how mushrooms are important to have in your diet, and people take them in supplements. But I think it's important to recognize that you don't have to take these things in supplements if you can just get them as a part of your diet regularly. And so if you were in a culture where eating mushrooms or eating corn smut was a normal part of your routine, maybe you're getting some of these important microproteins or other lipids or specific sugars that are only found in mushrooms just by eating them normally. And it's really interesting when we look at the history of corn smut. Its domestication history actually follows the domestication history of maize. And this makes sense, right? Because corn smut originally infected teosinte in the blue there, which is that small grain head. But very quickly, humans started to cultivate maize. And corn smut went through this bottleneck. It's this moment in evolution where something changes in the environment and only a select part of the population can survive. And that's because corn smut had to very quickly adapt to infecting a completely different shape of cob and completely different shape of, or completely different environment in order to best succeed. And so just as the maize was domesticated, the corn smut was domesticated by humans who were intentionally propagating it. And what actually was happening genetically was that the corn smut developed specific effector proteins, which are proteins that are secreted into the immune system of the corn in order to trick the corn's immune system and allow it to grow. And so here we have a three-way symbiosis, a really interesting relationship that humans have created through domestication, where we have a mutualism between humans and corn. We propagate the corn and help it grow. And then a mutualism between humans and the corn smut, where we purposely infect the corn plants and allow the corn smut to propagate. But within that relationship, we also have a parasitism between the corn smut and the corn. And one of the things that I think is interesting about this, and maybe has not been investigated, but I'd like to see it be investigated, is that by specifically allowing the maize to adapt to being parasitized over generations, are we tweaking the maize's immune system to actually be more resistant to other fungal pathogens? So are we developing crops that are better, are more responsive to fungal pathogens by exposing them to fungal pathogens over a longer period of time. So it's important to talk about why this actually happened in South America and Mesoamerica. Corn was domesticated in Oaxaca, and one of the things that's very fascinating about the Americas is that they are a huge center of diversity for plant and animal domestication. If you want to learn something about history, you should look at where things were domesticated, because it really clears up a lot of misconceptions about cultures and where foods that people eat actually came from. None of the plants that we eat today were domesticated in Europe. Europe didn't really domesticate anything. They didn't domesticate wheat, they didn't domesticate tomatoes, potatoes, or corn, or really any of the foods that are in traditional European cuisine. Actually, most of the foods that we are familiar with in the Americas and in the US today come from South America. So like I said, potatoes came from Bolivia, corn came from Mexico, tomatoes came from South America. And the reason that this actually happened was that there was a very diverse system of agriculture taking place in the Americas prior to colonization. People were growing gardens with polyculture systems that are very different from our modern agricultural systems that are monocultures. So they had traditional gardens where many things were being grown at one time. They had gardens where they were taking wild foods from the forest or from different environments and gradually over time propagating them, selecting them for the traits they wanted the most. And over time, that developed into a landscape where they had many different edible plants at their disposal. And we can see here in the top left that this is one of the ways that people are still growing vegetables in Peru to this day. It looks very different than if you picture American agriculture in the USA, where you imagine a flat field, one thing being grown. This is actually people cultivating things on hillsides and in valleys in high elevations. So very different than what we're familiar with. And then our second picture here, we have maize diversity in Peru, where we have all these different corn varieties that can be used for different things. Some are higher in protein, some are higher in fats, some are higher in starches, so they can be used for different foods. And what that also does is it also confers some ability for the corn plants to resist diseases, because if you have more diversity in your system and in your crop varieties, some are more likely to be resistant to a disease or a plague when it happens. Most famously, the polyculture that most people know is the three sisters, the corn, beans, and squash that grow together. And so that is, of course, all three of those plants were domesticated in South America and Central America. Another interesting thing to note is the terra preta phenomenon. And so this is something that took place in the Amazon. It's disputed among scientists if humans actually did create this soil. But usually in the Amazon, you have soils that are very infertile because of how much water gets moved through them and how hard it is to store carbon. But scientists have found these big tracts on the left-hand side of this image where you have very fertile, loamy soil. And one of the theories is that humans created this through some sort of technique of resiliently farming the land. And so you obviously had people, if this is the case that humans did create this soil, that knew how to work the land in a diverse way with a number of plants. And corn smut was a part of this. And right here we have an example of more domesticated crops from this region. We have the tomato, which was this tiny plant that was sort of poisonous to humans, and we turned it into the juicy tomato we know today. And then also the diversity of root crops and potatoes that are from South America. Another interesting one is milk, and fungi are really closely entangled in how milk has been used throughout history. So milk has been a staple food in Europe, and it's one of the best examples of selective pressure on humans. And domestication is a co-evolutionary process, like we said at the beginning. We are evolving just as the crops that we are domesticating are evolving. And so we domesticated cattle for dairy, we domesticated cows, goats, sheep, and a few other animals. And we've milked them ever since around 7,500 years ago in Europe. And one thing that happened was that humans originally were not able to digest lactose, and many of us still aren't able to. Humans naturally, when we're born, we have the ability to digest lactose, but only while we're breastfeeding. Shortly after that time, when we're weaned off, we lose the ability to digest lactose. But at one point, and this actually happened multiple times in human history, there was a mutation that occurred which allowed humans to digest lactose and produce lactase longer into adulthood. And so the humans that had this mutation survived longer and they were selected against, which is why in regions where humans were farming cattle for dairy, you have larger amount of the population that's able to digest milk. But milk back then looked a lot different as a food than it does now. Now, of course, we have refrigeration and pasteurization, invented in the 1800s for pasteurization. And this allows us to buy milk at the grocery store and store it for a week or two. But back in the day, milk fermented and spoiled really quickly. And the only reason we've been able to actually store it and use it as a staple food is because of the microorganisms that allow us to preserve it. So we can ferment milk using beneficial microorganisms into cheeses and kefir and other drinks. This gourd here, it's a bottle gourd from Africa. In some cultures that rear cattle in Africa, they store their milk, soured milk, in bottle gourds that they specifically select the bottle gourd for the shape that's best for holding milk and having it be the most convenient. So you just have a little pouch of calories that you can carry with you throughout the day. And they actually still grow these in Africa today. So with milk, you've either got to chill it in the refrigerator or you got to cheese it. And one of the ways that humans have done this is through the selection of very specific molds for making cheese. And so one of these is the camembert molds, penicillium. Who here has had brie before? Brie cheese, we all love it. But did you know that brie actually used to be blue? Brie used to be a blue cheese, and the reason that it's white today is that there was a mutation in one of the penicillium molds that produced brie that made the mold white. And over time, actually, it was not too long ago. I think it was only about 100 years ago that brie used to be blue. So over time, we've shifted which microbes produce our cheese products. And so the way that these are propagated is asexually, right? So you have a cheese, and in a facility, you have these molds and you add them in, and the spores that are produced there are re-added to the next batch. Originally, the molds that produced brie actually produced some toxins with them as well. So while they did preserve the cheese, they were also slightly carcinogenic and toxic. With our modern cultivars, if you see this phylogenetic tree here, the top there, that white brie is the most recent, and it's also the whitest. They do still produce a little bit of these acids that are toxic, but it's to such a small amount now that it's not really a concern for us. So over time, humans are selecting lineages of fungi that, on a civilization scale, are better for us health-wise, and probably taste better too. Real quick, we're going to talk about genetically modified organisms, which is a hot topic. Everyone loves to get upset about it, and we don't really have time to talk about it in depth, whether it's right or wrong, but I thought I'd show you some examples of how we could maybe use genetic engineering for fungi. And I thought I'd start out by showing you an example that if you're on the fence about GMOs, maybe this will change your mind. So transgenic yeasts, a yeast that has a gene from another organism that's been inserted into it that allows it to produce something besides alcohol. And so an example of this is rennet. Rennet is a chemical, chymosin is the actual chemical name, that allows cheese to melt. If you tried to make cheese without this chemical added to it, it wouldn't melt. So it's what allows you to actually get the stringiness in your pizza or any other melted cheese product. And the only way to naturally get rennet is to take it from the abomasum, which is the fourth stomach of a ruminant, when the ruminant is less than 8 weeks old. So these animals produce this enzyme when they're children, before they've stopped feeding from their mothers. And what this means is you have to slaughter young animals or harvest it from young animals and kill the animal before they're 8 weeks old. So if you're on the fence about genetically modified organisms, it might maybe help persuade you a little bit that even to just make the cheese produced in Italy every year, we'd need 4,000 tons of rennet to be produced. And that equates globally to millions of baby animals being slaughtered just for the production of rennet. So we can do this with a yeast by inserting a gene, just one single gene, to produce this enzyme in a big tank and then just filtering it out to get the enzyme. And so this is an example of a fungus that's sort of been domesticated to produce a product that's very important in our food system. Another use of genetic engineering that I'm a little bit less in favor of is the production of these sort of fake meat products. And The jury's still out on how people feel about these. I'm not really convinced they're as good of a food source as meat is. But what scientists have done, this actually is a paper that came out this year, is they've genetically engineered the Aspergillus oryzae, the koji fungus, and they've made it produce more of ergothioneine, which is an essential amino acid. And so it makes it nutritionally a little bit better for us. And they've also added heme, which is one of the components of hemoglobin, which is in your red blood cells, to change the fungus from white into this red appearance, kind of making it look more like meat. And so what we've done here is we've made a fake meat product with the fungus. Is it a whole food like a meat is? I'm not really so convinced. I don't think that you can just add one amino acid to a food and say that it is nutritionally complete. But as I said, the jury's still out, so we will decide if we want this in our food system. Okay, now we're going to get back to mushrooms themselves. And before we do that, I want to talk a little bit more about two important distinctions in the mushroom life cycle, which is asexual reproduction versus sexual reproduction. And so fungi aren't just like us. Humans can't clone ourselves forever and exist as duplicates of ourselves over generations. But fungi actually can do this. It's very contrary to the human lifestyle. We try and box fungi into human terms, but you really can't. They're so different from us. And so fungi can divide by mitosis, just like some of our cells can. And what you get is two cells that originate from one cell, and the final products are genetically identical. And so this is how our penicillium, our cheese mold, was dividing and being propagated. It's also how button mushrooms are cultivated and how many mushroom products that you buy online are being cultivated. So in a facility, someone has a petri dish and they grow it onto spawn. And then the same culture is being used and propagated and sent all over the world. So you have the same exact genetic copy of an organism being propagated. And this works really well if you want the organism to be very consistent and always be the same. But it also means that the organism doesn't really adapt to its environment very much, except in the case of a spontaneous mutation. A spontaneous mutation in the case of the camembert mold, where it became white and humans decided we like it white and maybe it's better for us, and we're going to keep that. But that's pretty rare that that happens. This is, of course, in contrary to sexual reproduction, which is where organisms go through meiosis, and one cell ends up becoming 4 cells that are each genetically different. And this is why when you have kids as humans, your kids don't look exactly like you. They look like a mix between the two parents, but also a little unique. And so the offspring's traits differ a little bit between each generation. And what this means for plants and for each different organism that we domesticate is that we can select the traits that we like the most. If anyone's ever had broccoli or cabbage or any other brassica like kohlrabi, those are actually all originally the same wild plant. They're all the same brassica called Brassica oleracea that humans have propagated and decided they want a really big bud or they want a really big leaf in the case of kale. And so we just decide what we want and keep propagating them. And each generation, we just select the trait we like the most. And so in domestication, this can be really extreme. And it's also what we've done with corn smut, right? So when people inoculate the corn smut plants in their field, they take the spores, the sexual spores of the corn smut, and each generation they're choosing the corn smut with the biggest galls that looks the most appealing. And this can get really intense. Broccoli is one of the most recent plant domesticates. It was domesticated in Italy. And you can see all these different modern cultivars and then also heirloom broccoli cultivars. They each look a little bit different, and it's a very human thing, right? Each different community was choosing which broccoli they liked the most for whatever reason. And I think that's beautiful. I think that humans creating this diversity of foods that's not just more nutritious, but also just really interesting to us is a really beautiful thing. But how do we achieve this with fungi? Well, if we wanted to achieve this with fungi, you would have to make the asexual propagation of fungi illegal almost. You would have to tell people that you have to select fungi, go through sexual recombination, and asexual propagation is illegal. And we actually did this. Not intentionally, though. In 1970, Richard Nixon enacted the Controlled Substances Act. And what that meant was that psilocybin became illegal. What that also meant was that there emerged this huge trade of psilocybin spores because of the reasoning that the spores of psilocybin-containing mushrooms themselves do not contain psilocybin. So through an interesting legal loophole, the spores of psilocybin-containing mushrooms were legal, but the mushrooms themselves were not. And so there became this big trade of mushroom spores on the internet, and originally just through word of mouth, and people started cultivating these things. And right, when you go through spore, you're going through sexual recombination with fungi. And so Richard Nixon said he will make psilocybin illegal. And what he did was from 1970 until now, he created a huge domestication of psilocybin-containing fungi. The war on drugs was not really working in the case of mushrooms. I think it's at the start of David Aurora's book. He gives the acknowledgement that he thanks his mom and dad for telling him not to look at fungi, and that's what made him love mushrooms. So it's the same thing here. Psilocybe cubensis, or just cubes, or magic mushrooms that most people know, have all these different morphologies. Some are albino, some are huge, some are tiny, some are really potent, some are not. And I think this is a very interesting case study. I think it's one of the most unique examples of mushroom domestication that you could find. And that's because while you could look at this and say that people are domesticating these things for money, maybe that's part of it. But I think the bigger picture is that humans really care about these things. We all care about mushrooms, obviously, if we're here, but a lot of people care about magic mushrooms because we have some sort of attachment to them from the experiences that they give us. And so this is humans, in a way, interacting with them and selecting traits that they see as being beautiful or appealing in them. And so I think in this conversation about growing mushrooms, it's important to cultivate mushrooms that are native to your area and cultivate mushrooms that you can actually interact with where you live. And there's multiple reasons for that we'll get into. But one case that I just can't help but bring up is the case of invasive mushrooms or fertilized mushroom populations. And so currently we grow in facilities a very few number of mushrooms, and usually the ones that we grow are because they have a flashy color, they're pink oysters, blue oysters, something like that. And this yellow oyster that you see a lot of people growing or being sold in grow kits is not from the US originally, and it's become pretty heavily invasive in the eastern United States. And so you see these trees that break out into these huge flushes of this mushroom. And when we think of decomposition of wood, it turns back into soil, but you want that to be with fungi that are releasing as fungi decompose wood, they're releasing gases into the atmosphere, and you don't want it to be too fast. It's just important that things happen in the natural environment with their native organisms for a variety of reasons. And so we know now through genetic evidence that this golden oyster mushroom was introduced from many different introduction events. So many different people introduced it, but there were only a few actual original cultures that were used. And so that tracks with it being from grow kits or from just a few companies. And so one of the other big questions with this is, could it hybridize with our native Pleurotus species? And actually in the lab, they've shown that it probably can. And this leads to the concept of something called outbreeding depression. So you've probably heard of inbreeding depression, which is the phenomenon that if you breed things that are too closely related, like in the case of humans with one another, like in a royal family, they used to do that in the UK, you get traits that are maladaptive and the organism can't really handle it. But it's kind of the same thing if you breed something that's really far away from it. And that's because if you breed two species that aren't really from the same place, maybe you inherit traits that just aren't good for your environment, right? The traits that make the golden oyster good in the old world or Asia aren't going to be the traits that make an oyster mushroom adapted to its environment in North America. And so in the lab, they've shown they can hybridize. I haven't seen any evidence of someone saying that they were actually hybridizing in the wild, but maybe it could be happening. And so that's why I think it's important that we just focus now as we possibly can on domesticating our native fungi here in the Pacific Northwest. And so some people that I like to point out that are just doing this in the USA are this guy, Jacob Alvarez, who got really into cultivating beefsteak mushroom. And he cultivated all these different lineages of the beefsteak mushroom, which is a pretty uncommon mushroom, actually. It's a pretty cool mushroom when you find it. And he collected all these and cultured them. And this is really important because the forest where he found all these beefsteak mushrooms was actually cut down and turned into an Amazon warehouse. So all of this diversity is now gone And he was the person that actually preserved it through culturing it. And you can do this too. He also did it with white-pored chicken of the woods. And this is a great mushroom. It's one of the best tasting mushrooms I've ever had, much better than normal chicken of the woods. And it's really interesting to me how each of these collections has completely different phenotype. They all look very different, right? Some of them look very brain-like, others look very fan-like. Sometimes the ridge, it's a very stark difference between the white and the orange. And so this is almost like the case of the Psilocybe cubensis, where we had this huge variety of traits and colors, and humans are choosing to propagate the ones that we want. And I really think we could do this, and just if we all made a conscious effort to grow mushrooms in our environment, we could choose ones to us that just look more beautiful. Another person that I like that's doing this is Ryan Paul Gates. He's really big on collecting native Ganoderma and actually Ganoderma from around the world, and cordyceps as well. He grows cordyceps on eggs. It's just really cool. And so in my own initiative here, I'm trying to do this in the Pacific Northwest, and I'm fortunate to work in a laboratory that has a large culture collection of native fungi. And so we are propagating hundreds of species of fungi, and I'm trying to cultivate ones that aren't normally grown to see which fungi we could possibly domesticate, and some for food, others just selecting them to look cool or to be larger or to have interesting traits. And so our collection is pretty diverse at this point. We have over 50 families and 300 plus species, and it's constantly growing. This is at OSU, and we welcome people that have interesting fungi that they'd like to add to the collection. I can culture it. Or if people have a fungus that they want to access that's in our collection, I'm happy to hopefully make that happen. And taking this to the next step, I kind of wanted to get even more local, just as a kind of a treat to myself. And so I'm always looking at where things are wasted in our food system. And OSU has a pretty good barley breeding program that produces a lot of waste barley. So barley has a pathogen that grows on it called smut. And if the barley gets infected with the smut, you can't replant it. And so you can't really eat most of it either. What you can do is you can autoclave it and grow fungi on it. And so I've been doing this. I take all their waste barley, and that's what I grow the native cultures on. So my goal is to take native cultures, grow them on the local barley, and then use them for mushroom projects here in the Pacific Northwest. So just trying to think of not being influenced by growing mushrooms from companies. I don't think there's anything wrong with that, but I'm trying to make it as local as possible. Another thing that I've been trying to do is just trying to look at how people are growing mushrooms like corn smut in Mesoamerica and trying to incorporate them into my own gardening and farming. And so growing things in polyculture systems, this is a variation of the Three Sisters garden. But as you know, in the Pacific Northwest, we have very dry summers where we don't get much rain or any rain at all these days. And so I'm using crops from Africa that are adapted to very dry summers. And those would be sorghum, bottle gourd, and cowpea. And this works the exact same way as the Three Sisters garden. The reason I grow the bottle gourds is because I've figured that we can kind of use them sort of like how people use grow bags to grow mushrooms out of. And so this is good for a variety of reasons. I think it's really cool just from a scientific point of view because contrary to popular belief, wheat is not the first domesticated plant. The bottle gourd is the first domesticated plant. And it makes sense, right? Before humans were domesticating things for food, they were just domesticating things for utility. And also, in nature, these just look like little round balls that are maybe fun to throw around. So maybe humans were just carrying them back to their settlements and then throwing them around and unintentionally propagating them and selecting the ones that were the roundest containers. And so you can dry these out and they form hollow containers that are pretty hard. Another thing is that prior to wood turning and other materials that we use today, having a hollow shape that can hold water is pretty difficult to make. And so if you can find a plant that can do that, it's pretty significant. And also, bottle gourds have a white flower. They're moth pollinated. So if you're just thinking from a pollinator point of view in your garden, it's cool to have a plant that's moth pollinated at night, also to see some cool moths. So I've been trying to just stuff these with different substrates like straw and grow mushrooms out of them. So growing local mushrooms out of the things that you grow in your garden, if it works, great. And if it doesn't, you can just compost it. You didn't use plastic. This conversation gets a little bit more complicated when we consider that we don't always know which mushrooms are native or not. It's not really a topic that's been explored. And this is a really cool paper that came out a couple years ago that showed that it confirmed a theory that's been around for a couple years, that the native psilocybe that we have in the Pacific Northwest might not be native after all. And I'm talking here about psilocybe cyanescens and azarescens specifically. So these are the fungi that people talk about going to the dunes or going to dune grass to pick. And what this paper suggests, and pretty convincingly suggests, is that these mushrooms are actually invasive mushrooms from Australia. So the theory goes that there was this huge trade of forest products from Australia to the US during the 1800s. And back then, we didn't have biocontrol. There were many of these ships that crashed along the Oregon coast, and they had timber products that carried mushroom spores. One of those mushrooms was Psilocybe subaeruginosa. And as it crashed along multiple places on the coast, these mushrooms escaped. And the subaeruginosa, being exposed to a new environment, went through what's called allopatric speciation, where you have a mushroom or an organism that arrives to a new environment, and because it adapts to its new environment, it goes through all these changes and becomes its own species. And so that's what we think happened with cyanescens and azarescens. That's probably why they look so similar to each other, except you have very small differences in the cap shape. And I think it's an interesting conversation because obviously we don't want to introduce invasive mushrooms, but we have these invasive mushrooms that are already here, and humans clearly care for them. In Oregon, there's this big culture around these psilocybe mushrooms. So do we make the exception for these mushrooms and say that we care for them and we want to keep the cyanescens and the azarescens, or do we try and eradicate them? And I don't think we even could eradicate them. So open debate of whether it's good to have these invasive mushrooms around. Now we're going to talk very quickly about just some of the more fringe applications of could we do this? Could we domesticate fungi? And I want to point out that lettuce used to be a pretty toxic plant. You can still find these wild lettuces growing as weeds. And if you rip one of their leaves off, you'll see this white, milky latex that comes out. And that actually contains a compound called lactucin, which has an opioid-like effect, but in large amounts, it's poisonous. And humans could eat this in very small quantities, or they could kind of leach out that toxin by cooking it. But over many generations and many thousands of years, we domesticated lettuce to not have this bitter compound anymore, or just have very small amounts of it. And so we took a plant that was too bitter to eat, or sort of inedible, and we domesticated it to be edible. So could we do this with fungi? Could we take this giant laughing Jim and somehow select it to become an edible mushroom? I don't know how we'd go about doing that, but maybe we could through conventional breeding, or maybe we could do it through CRISPR. I think that's a bit more of a stretch, but just theoretically, could you take a poisonous mushroom like an amanita and remove a gene that makes a toxin and make it edible? I don't know. Sometimes when I research domestication, I'll come across these clickbait articles that say, this is what watermelons used to look like before people domesticated them, before people changed them. It's actually not what watermelons used to look like. Watermelons actually used to look like that, and they were just a few centimeters across. The pictures that you see online are actually what watermelons looked like after thousands of years of human domestication. And the difference between that photo and that watermelon that we have today is only a couple hundred of years. I think that photo is from like 1600. And so this is kind of my big theory of maybe where mushrooms are at today. Maybe we think we've domesticated them, like our button mushroom or our portobello. We just haven't cultivated them enough. Maybe we're at the place that the watermelon was at a couple hundred years ago, where we're thinking, this is as good as it's going to get. But with a couple generations of human life, if we really cultivate these things and take care of them and select them for what we want, maybe we could produce something that looks entirely different and is good for us and allows us to engage with fungi more. And I couldn't finish this presentation without talking about a very, very unique example from nature. It shows that humans are not as significant as we think. And it's the case of fungus farming ants, atine ants. And this is a symbiosis that started around 65 million years ago. A paper that was just published about a month ago showed that this symbiosis actually formed possibly after the extinction event that killed the dinosaurs. And so what scientists think happened is after the asteroid struck the Earth, it caused what's called an impact winter, which means the Earth gets covered in a cloud of dust. that stops the temperatures from rising very high. And it meant that photosynthesis was pretty much halted or significantly reduced for a number of years. And that meant that there just wasn't that much living biomass for things to feed on. It also meant that if you could feed on dead plant material, you'd be a really successful organism. And the scientists proposed that the ants saw this and gradually found that they could cultivate fungi that were already around in soil and domesticate them and actually feed on them. And so that's why you have leafcutter ants today that you've seen in videos in the jungle that cut down all the leaves in the jungle and they actually take them back to their nest. They cultivate this fungus. It can actually be a few different types of fungi, and there's over 250 species of these ants. But they have domesticated this fungus to actually produce different structures. So microscopically, these fungi now produce these filamentous heads called ganglidia that are really nutritious. So they grow the fungus, they allow the fungus to survive, and then they eat these swollen heads of the hyphae. So I think if ants can domesticate fungi and fundamentally change them on a microscopic level, we could too. We just need to invest more time into it. So I want to recenter this presentation and just bring us back to our timeline of human history. Humans have only been around 200,000 years, give or take. We've only been domesticating organisms 15,000 years. We've only been growing mushrooms about 1,000 years. And really, we've only been cultivating mushrooms with our current scientific knowledge of fungi for about 100 years. So if this is where we're at with mushroom cultivation today, I ask you to consider what comes next. If we really try and grow mushrooms and domesticate them and select them for our environments, what can we truly grow? With that being said, thank you very much. Please feel free to reach out with questions. I've got a bunch of squash and other plants over here if you'd like to support what I do. I'm doing a couple research projects at OSU, and those are made possible by donations and organizations like this that allow me to speak, and also through people that buy my produce that I farm. So thank you very much.
You have questions for Charlie?
How different is the process of domesticating fungi versus plants? There's not a huge fundamental difference. I would say the biggest difference is that the body of the fungus, as you know, the mycelium, is usually invisible to us. It's not like a plant where you can see it living above ground. And so if you want to propagate a fungus and domesticate it, you need to be selecting the best lineage and then propagating only that one. And so that would mean just choosing the biggest mushroom and then cultivating it in a different space where it's not growing before. And so I think that's probably one of the reasons that we haven't domesticated mushrooms before, is because we can't see them most of the year. But in terms of biology, what does it take to select them? It's about the same. And the best example of that is the magic mushrooms that we talked about. Most of them are grown indoors in big warehouses that are climate controlled. You can look at Asia and specifically China as a country that has a much more diverse way of growing mushrooms, like growing reishi and many different types of mushrooms in greenhouses. And each mushroom farm has its own way of doing things. And that's just because they've been growing mushrooms a lot longer than we have in the US. And so they have some cultural adaptations of how they've chosen to grow mushrooms. But I would even say that the way that we're growing mushrooms in Asia, it's still mostly the same clonal lineage being propagated, and they're not breeding them. And for context, the main button mushroom variety that's cultivated today in the US, it was bred in the 1980s. And I think there's been one hybrid release that's cultivated since then. But they just don't make changes that often. And the main reason for that is it's kind of like a if it ain't broke, don't fix it model, where there's only two big companies in the US growing button mushrooms. So no one's going to outcompete them. Why would they have to change anything?
We have an online question from Jessie Ewing. Do you know her?
I do.
She's asking, what traits would you change, enhance in your favorite or desired edibles?
Okay, what trait would I change in my favorite edible mushrooms? I think if I could, I would select for smell. I think if every mushroom could smell as strong as matsutake would, that would be amazing. I think another trait that would be good to work on would just be spore production. Sometimes I think about when you see mushrooms in the grocery store and they have like spores dropping onto them, it kind of looks gross and the mushrooms don't look as good, so they don't have as long of a shelf life. So that would be one I would work on. And maybe make the mushrooms non-sporulating. It's kind of like a seedless watermelon.
But as mushrooms mature, doesn't that sporulation actually change the flavor of the mushroom?
I'm not sure.
Ooh, that's what I thought.
Well, yeah, it does, because if you're saying the Garicus bisporus, the button mushroom is the same as the portobello, they have very different flavors.
Right.
So I think the spore production or the maturity, or when you get spore production, probably leads to more diverse flavor.
Right. Yeah.
Other questions for Charlie? Did you want to talk about your squash collection here?
Sure. Yeah. I've got I don't know, 5 or 6 types of squash here. I've got these giant North Georgia candy roasters. Everything's name your price on them. And we've also got some Oaxacan green corn you can take home and make masa with, or just corn flour. Yeah, everything's name your price. We got spaghetti squash, and then we've got 2 pie pumpkins that are great for making pumpkin pie in time for the holidays.
Thank you, Charlie. Next month is our invited speaker is Dr. Biddy Roy, and she's going to give a presentation on the macrofungi of Lane County project that's been going on for 4 years now. So stay tuned for that. Thanks for coming.